The gut microbiome is a collection of microorganisms that is inside the gut of the animal. These can include bacteria, bacteriophage, fungus, and viruses. When people are in the infancy stage and the old stage of their life, immunity is at its weakest. Intestinal changes occur in the microbiomes. Microbiome increase as the host grows older. Our microbiome and aging are a correlative and normal relationship that you cannot control happening. And the relationship between gut microbiome and obesity is the composition and function of the gut microbiome. Obese people have more firmicutes and less bacteroidetes than lean people. The more caloric intake is increased by the firmicutes grow by 20% and decrease bacteroidetes by 20%. The relationship is correlative because you can only analyze the correlation of data. You can't do a controlled experiment with people because that's not ethical. For our hypothesis, a 12-year-old snake and a 2.5-year-old dog will have differences in their gut microbiome, but since both have a high-protein, all-meat diet, there should be many similarities also. We examined samples from a pigeon, iguana, and two different dogs, a pit bull and a chow, and the good results came from a husky and a corn snake. Purification of DNA from fecal matter was performed with the Quigen column, and DNA was isolated from the fecal samples and increased tenfold. Using the Zymo column, the concentration of DNA was increased while the sample volume was reduced. This was done per manufacturer's instructions. In order to determine the concentration of DNA, a UV spectrometer was used. An nanodrop, a special type of UV spec, was used because it only requires a small volume of sample and it automatically calculates the DNA concentration. For gel electrophoresis, uh, different molecules are separated based on their size, and you can see our results for that right here. During the PCR cleanup, the use of the Agincourt Ampere kit, the amplicon will be cleaned up from the amplification PCR reaction. This cleanup will remove the components of the amplification PCR buffer, as well as unused forward and reverse primers. Indexing PCR will, in order to determine which sequence came from which scat samples, a unique set of short sequence tags are attached to the ends of amplicons from each sample via indexing PCR reaction. In this experiment, only the 3' portion of each primer will attach to the amplicon. The 5' end of each primer will contain the unique sequence tag needed to identify the sample. And only 8 cycles of this were ran, and this is our results for that. This is the results of our good microbiome data, analyzing the kingdom, order, and genus of the microbiome of a husky and a snake. At the kingdom level, all samples represent similar readings, mostly bacteria with trace amounts of other or non-classified organisms. The native control had a very small number of total reads. At the order level, differences emerge, but there are also similarities at every level. The positive control shares the strain Enterobacterialis with our sample for the snake, but more similarities exist between the two test samples, specifically Clostridiales and Bacteriodiales being among the most prevalent orders. At the genus level, the same for strains. The most abundant strains are the same for both samples. Bacteroides, Glaudia, Clostridium, and unclassified, but all occurred at different proportions. Positive control contained mostly Escherichia, and negative control was mostly unclassified. So the gut microbiome for the 2.5-year-old husky and the 12-year-old snake were similar, with some distinct differences. The highest occurring bacteria in both organisms are found in the gut and participate in digestion, although they do in different ratios. This can be due to differences in diet, metabolism, and environment. Presence and quantity of trace organisms was different, although their role was less clear. The major difference between the two microbiomes was the presence of E. coli in the snake, but not the husky. This could be because the snake eats whole mice and could have introduced E. coli that way, but it also could be because of an error in tagging. The number of unclassified reads could easily contain Enterobacterius bacteria, and there really should be E. coli in both organisms.